Hello, everybody, and welcome to today's class where we're going to talk about Nextflow, installing Nextflow, and what the design file Nextflow requires. The previous video went over what Nextflow is and how we're going to use it, and we'll touch on that a little bit as we go through the document. But today we are on 05, Nextflow install path and trial design. So if you click on there, you'll see all the stuff we need for today. And so the first thing we need to do is install Nextflow. And Nextflow is going to manage the container, essentially. We're going to develop a container with Singularity, and then Nextflow is going to put in all of the programs that we would need. And we'll go over what those programs are and output files in later video. But for now, we want to install Nextflow, and so you can read the Get Started markdown or materials here. It's really very well done, and also for ChIP-seq, which we will install in just a little bit. But first, we want to install Nextflow, and so I went to my home directory here on this terminal. And you can go to your home directory. I recommend going somewhere upstream, if you will. And then to install Nextflow, there's this one line here. It's really simple. It's remarkable what they've done. And so what you're doing here is you're doing wget, which we've seen. We'll go get a file. And the Q is for quiet, and then the O is for output. And so it's not going to like show you everything it's doing and print it to the screen. It's going to send it from this website and then into Bash. And so Bash will then go in and install all of the files needed. So it's really that simple. And long ago it was much more complicated than this, but we can see if you go like this, you are going to get Nextflow installed, and we'll let that run for a minute. It's already installed, and so we can see that we installed version 20.1.0, and as of the filming of this, that's the most current version. What's interesting, if you do have older versions of Nextflow in your, if you've already done this before and you go and you look, you'll see that we have a .nextflow file now that we've installed it. And if we look in there, what we can see is this thing called framework, which is which version it is. And I have 3 different versions installed in here. And so that's fine. And it's good to have the other versions if you want to call them back. So yeah, so that's all that was to install Nextflow. And when you list, you should see now a .nextflow file here, folder, sorry. Okay, so great. Now we've done that. Now we want to discuss path. This is a sort of an esoteric concept, but it's really important. Imagine that when you run list or cd, that you had to tell the computer where that command was. And so you had to type out blah, 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 /list to call that every time. That would be really a lot of work. But if you put something in your path, the computer's going to go there right away and look for what commands are in there. And we have an exercise below that will show you sort of the importance of path and how powerful it is that if you write a bash script that does something like collect all your photos from iTunes library, which has got a bunch of folders in it, you can run that from anywhere if you put it to your path. So this is going to seem like a little bit of a detour, but I think it's important to get this concept sort of down. And so what we can do is use echo to just see what our path is. And we'll have a path file come up here, and you can see it's kind of hard to read. It's got all this, you know, / and it's a colon delimited file. So what we're going to do is use transmute. This is a very powerful bash function, which is sort of like find and replace. So you can change the colon here. It's just going to say transmute. What do you want to change? What are you looking for? And what do you want to change it to? And we'll just make it a new line. We'll use transmute again later. but it's a really handy way in Bash that if you're not certain about if you want to make sure it's a certain file type, you can change the delimiter really quick with this. So if we just now type all this out, we get a much more user-friendly sort of path. And what you can see is that I've told the computer that, you know, Nextflow commands are in here. And these are my older versions of Nextflow as well. But it's going to tell it to go where to go there. And for list and things like that, it's going to go into your more user local bins and sbin. So this is really handy. And you'll see here we have this time script exercise where you can follow along. We won't go over it in this video, but if you just follow along with this markdown file, you'll use some bash, which is fun. It's has markdown commenting on what's going on here, but you can see transmute being used and cutting the fourth column, which is the time, the actual time. And you can go through this bash exercise line by line. And what you'll do is you'll make a command that you can be anywhere. And now I can do, since I've already run this, I can run what time is it? And this is what the script will output. It's 5:35. I'm glad to see you're making use of it. Okay, great. But the cool thing is that you could do something much more powerful than this exercise. And then that way, when you set it to your path, you can use that command wherever you are in whatever directory. For example, this is handy when I want to grab pictures from a certain year or month. I can easily use this instead of having to go through iTunes and pick them out one by one. Okay, so now what we want to do is actually put Nextflow into our path. And so that's here on line 187, where we're going to echo, which is just going to tell us what the path is. And we're going to put it to where it is. And this is my user. When I go to here, when I go to here, I can see this is that user's Jory 2700, and that .nextflow folder is in here. This will be different for your user and ID. And then what we're going to do is output this to our .bash profile, which is in our home directory as well. Okay, so then basically what we're just going to do in summary is echo to our path to export a path to this, to make this into our path file, basically. So let's run that. Okay, so now that .nextflow should be in my path, but as you can see, it already was installed. So, but let's make sure it's there. We'll just echo it and then pipe it to transmute and change the colons to a new line just to make it easier. And now you can see that .nextflow. And you'll do that for each version you install. It'll make the same entry. Okay, so there now we have Nextflow installed, and Nextflow knew where to put everything, and now we can be in any folder and call up Nextflow like we do list or CD. So that's just sort of an example of why path is important to know, that it's a handy place to put stuff that you're going to use, that you don't have to call up the path to invoke that command or that language. Okay, so now what we'll do is install the NF-CORE ChIPseq pipeline. And this is even simpler. Look at this, they have one line and we will get this entire pipeline that has a genome alignment algorithm in it, a peak caller. It's going to produce tons of files that are very useful. And so by just pushing fast queues through this pipeline, we're going to get a ton of data, and this is all it takes to install it. This is what you would do to install the latest version. So when you can see here, it'll go to Nextflow, and what it's actually doing is it's going to the Nextflow GitHub, and this pull command we learned in the Git lecture, that pull is going to go now pull down this entire repository and install it for you. And so there, when you run this, you will see that, and then if you want to you'll see a bunch more happen. I already have it installed, so it's saying it's done and it's up to date with 1.21, which is what we want. And then, or you could specify the version. Let's say you wanted to go back a version, you could do 1.0.1 here. And then actually just, and if you want a specific one, you can specify this. This actually needs to be corrected, that this flag 1.21 should be after pull. I'll fix that. Sorry, just noticed it now. Okay, so the good news is you can install the latest version. But when we go to see how we're going to run Nextflow, we're going to have to give it a few files, and one of them is the run file. And that's going to tell it what version it is anyways, and it'll just install it or check to see that it was installed, like this one did here, and said it's done, and it made this new revision key. But if it's not installed, Nextflow is going to do it anyways from your run.sh file, which we'll get to in just a second here. Okay, so another thing Nextflow needs is a configuration file or a config file. And this is simply saying to your server or your computer what needs Nextflow is going to have. So the needs it needs is how it's going to talk to your server or computer, and that would be through Slurm. And then the queue, we're going to go in the short queue so we can process fast. In memory, we're going to ask for 32 gigabytes of memory. That's plenty. We probably don't need that much. And then max forks is the amount of jobs that can be run in parallel. So let's say genome alignment is the first process. If you have max forks of 40, then you can have 40 FASTQ files being aligned at once. That's kind of heavy on the server, and you should talk to your IT people about what to do with this. In the class, we'll probably turn that down to 10 jobs in parallel. So you're going to need this in a text file, and it's going to be called nextflow.config. And Nextflow needs that in order to talk to your server and allocate the resources to Nextflow. And you'll see that, well, in Slurm, so it's a good transition. Here we are, Slurm. Is how you're going to manage the jobs as they're going. So Nextflow is a package manager, and this is now we're going to do a job management. We're going to tell what scripts to run, send them in batches to Nextflow. And so here's an example. If you've cloned this GitHub, you already have a run.sh file that's fully ready to go. But here we just have markdown of what's going on at each step. And you'll see here that for Nextflow configuration, we ask for the short queue, but with batch sending just basic scripts every here and there, now and then, it doesn't need to really be in a fast queue. And it also doesn't need that much memory, as we'll see in a second. But you can also give it a job name with these flags. The mail, you want to send a mail message if it fails, and what that email is. Then you'll ask for how many nodes. You'll usually only want one node. Our server has multiple nodes, but nodes aren't very good at communicating across each other to resynthesize the jobs. So typically it's best to stay on one node. End tasks is essentially how many tasks is Slurm running at once. And in this case, we're only running one. We're running this run.sh file. Now let's say we were running two run.sh files, then this would cause a problem because it's saying, Slurm is saying, well, I have two jobs to do and you told me only to do one. I don't know which one to do. But that doesn't really matter. You'll just set it to one. And then the memory here is 6 gigabytes. Again, we don't need as much memory because we're just organizing the jobs to keep moving. This is the wall clock. This is an obnoxious 100 hours. That's what it will take to run this data set. In the first test run we'll probably set this to 5 hours. It's good manners to try and estimate the wall clock time with the actual time you think it's going to take to run, because the server has to manage a bunch of jobs with a bunch of people. And so it kind of wants to know when it can plan to stop your job. Okay, and so we're going to tell that we want to print out the out. So we saw before that you could use the arrow or the forward arrow to make an out, so you could see what the computer's printing out as it goes. And we also want the standard output, and we want the standard error. So we can follow along, and we'll see that in just a second. Okay, so that's Slurm, which is going to manage all the scripts and things like that, whereas Nextflow is going to ask the server to have its own set of resources to do its job. Okay, and that's marked down more in here. Okay, so now the second half of the run.sh is for Nextflow. It's telling Nextflow the things it's going to need to know. So it's going to, you just want to check where you are and put in some text you want printed if you even want that. So this is a bash command to load Singularity. And in the video associated with this class, we go over what Singularity and containers are and how Nextflow is managing them. But essentially you can think of it as an empty space where Nextflow is going to say, okay, load this Singularity container, and I'm going to go put in all the programs I need into it. And the nice thing about this is that it's 100% reproducible because all the right versions, all the right things are documented in this container. And the next lectures we'll talk about what those processes, all those programs that are laid into the container to make it into a pipeline. Okay, so you can see I called up an older version. We're test running this for now on the same version we used last year, but you saw that we installed 1.2.1, so I'll probably change that before the actual class. We're setting the profile to Singularity, and that means that Nextflow is going to use a Singularity container. You could set this to a Docker container or other container programs you want to use. Single end here is required. If you have single ends, it will default to double end reads if you don't put this flag in there. This is all in the documentation, which is in this markdown file, but I thought it'd be worth just talking about the key ones. So one thing we need here is a good input design file, and I'll show you an example of one in a second here, so you can actually start running your first ChIP-seq at the end of this video. But you need a design file, and we'll talk about what that is, but Nextflow needs to know what files are which ones are your controls, and which ones are your test samples. We also need the human genome. We have to line these sequences from this data to the human genome. So in class, we have a file path here that everybody can get to where that is stored. And so Nextflow is going to look for that and say, oh, here it is. Okay, I'll go there. Similarly, we need an annotation for the human genome. That's in a GTF file. Again, we'll talk about that in the next lecture, what all these file names are all about. But you not only want to align to the genome, you also want to keep track of information as where is a gene, where's a promoter, where's an enhancer, and things like that. Max is one of the programs that Nextflow is going to run to call your peaks, to say where the signal is, the kind of file you want. And Max requires a parameter about how big your genome is. So right now we're doing the human genome, so it's about 3 gigabytes. But if you were using a smaller genome, it would need to know that. Like if you're using the yeast genome, which is only about 12 megabases. So, okay. Then you also need a blacklist file here, which is all the bad regions of the human genome that we don't want a Nextflow to align to. It's going to call up BWA. We'll go through that again in the next lecture, but it's going to call up BWA and it's going to say, hey, don't align to these regions. They're unreliable. And then this is kind of standard stuff of your email, having Nextflow send you an email. So you get one from your server and one from Nextflow if you've been successful or if you've failed. And resume is a flag in Nextflow so that sometimes these pipelines can run for days and you can resume if there's an error using this flag. If you don't put this in there, it won't allow you, it'll just cancel the entire thing. And then also to call up the Nextflow configuration file. So you'll see this nicely laid out in the run.sh file, but I thought I would just go through what each of these sort of lines are. And again, if you go to the ChIP-seq NF-CORE documentation, it'll tell you about all of these things in much more detail. And that link is in here as well. And I just note that each command for Nextflow is ended by one of these backslashes. So this is all, everything I just said is also written out here. If you want to get the GTF annotation file, that's shown here. Everything's laid out in more detail. But when you're running a job, you're often going to want to check on it. So instead, I thought we should go over how to check on your runs before we actually run something and need to check it. And so this is what we're going to actually run. Don't run this yet, but that will run our run.sh file and instigate all of Nextflow. And that's what we'll do at the end of this video in just a couple minutes here. So if you want to see what's going on, you can look at SQ in your server. So SQ and then -u is for user, and my username is Jory2700. And you can see I have no jobs running. And we'll check that again after we start running a job. But sometimes you might see here in this job ID that you have like 20 jobs running, and you don't want to go do that each one by one, where you say S cancel, and job ID is the way to cancel a job. So you can see what's running, look for the job number and cancel with that. That can be quite a problem if you have a bunch of jobs running. But if you want to cancel them all just blanketly, you can use scancel your -u and then your username, and that'll just cancel everything that's running. A little more elegant solution is here. Don't be too scared by the bash, but I think hopefully it's starting to become kind of something you can see and think through, that you're gonna type out all the jobs you have running, and now grep is gonna go for the job ID, the common job ID number. So like if it's 597 for all of your jobs, then you would use that number. awk will print the first column and then send this into the shell memory to do scancel one at a time. And so then that way you could use grep to just grab like 7 jobs that you were interested, or subset using regular expression. Probably won't need it, and usually you just would cancel everything if you knew you did something wrong, but just in case, this is outlined as another way of things. So you can cancel all your jobs, great. We want to do that before we run because we're going to cancel them pretty quick. So what we need, last thing Nextflow needs is a design file. And these are all the components of that design file. The group is essentially the gene you're interested in. If you're interested in, we're going to do WDR5, which is changes the histone packaging material DNA to have different modifications. That's a protein we're interested in called WDR5. We might have multiple replicates, so we would have an entry for each replicate. The FASTQ, since we're only using single read data from this ENCODE download, we'd only fill in the FASTQ for one and leave this one blank. But if you do have paired end data with RNA-seq or other types, you'll use both ones and you'll tell the file path to them, and you'll see that in a second below. The antibody, which is really the same as the group, so in this case it'd be WDR5, and then the control. So this is important, that this is usually the input DNA, and in the next lecture we're going to talk about a little bit more about the aspects of ChIP and what we're looking at here. But the control is essentially generic DNA that if you're suspected that your protein binds in a specific site, that the chip would have more signal there than the control input. But the control input also needs to be entered as a group. And so we can see that in a second here. If you want to do this, these are also mentioned, everything I just said is mentioned here. If you want to do this along with us, you can just download these with wget. These are the only 3 files you'll need. We're just going to do this very simple first test run, but you will run your first ChIP-seq right after in just a few lines here. So here's an example of one where ASH2L is this WDR5 gene I was talking about, and we have our first replicate. Now this is going to be important for you to figure out where your FASTQ files are. Where I'm going to be working, this will be one directory below where the FASTQs are. And so I'm telling you to go up a directory, look for this FASTQ file for that one, and the second one is empty here. And then the control or the group, or sorry, the antibody again is very similar to group. And the control is this number here, which you downloaded here. Okay, so then this control needs to be added as an entry as well. So it's added here. It's only gonna have one replicate for the control, and it's in this same file path. So you do need to add this, and then when the control, you don't have a control. You leave that empty. So there's a couple little things to think through. This right now is tab separated. It needs to be comma separated. So I just thought you could try and do this real quick and just see how you can easily use transmute to change this from tab here to comma, and voila, you get this, where we have ash1 is the group, or WDR5 ash2L, replicate 1. Here's the first FASTQ file path. And now you can see the commas are empty. The group, or the antibody is ash2L the same, and there's the control. Now you can see the control file is empty for the control. Okay, so once you have all that, we're gonna go, let's do a run. So let's see where we are. Let's go to class. Okay, so we're gonna go into test work just for now. Go into whatever directory you want, and I'll show you what you're gonna need in there. So in here, we'll have listing all the things you need, but just to highlight them, you're gonna need a design file as a CSV, and that's this shown here. We're gonna need that Nextflow configuration file. You can just add that in nano and copy and paste from this RMD file. And Nextflow out and Nextflow error are gonna be produced. I've already run this, so those won't be there for you. But you'll need your run.sh and your blacklist. So once you have all these, and again, it's outlined in here if you want to go back and read that, but we can see this run.sh file now in its full glory that we just went through, all of these things that it's going to need. And so when you want to run a shell script, so you can see that run.sh ends in .sh, that means it's going to try and run on the shell. And so since we are in on the server and we're going to use sbatch, we'll do sbatch run.sh. So that's here. It's just that simple. Because we set it up so well, we're just going to do that. And we're doing ChIP-seq analysis right now. And Nextflow already knows everything we probably want to know and is making all of the files for us. But how do we check in on it? Well, let's look in. We can do tailf -f. That means follow the Nextflow output. So that's going to say where Nextflow currently is. And so here we can see what it's doing. And the nice thing about the -f is it's going to update us. And we're going to go in the next lecture what all of these programs are, but you can see it's doing all of these things you would have had to do one thing at a time. First, it checked our design file. Then it's looking for the to align to the genome. It's going to do some QC on that. It's going to give us bigwig files that we can use in the browser. It's going to give us peak files. It's going to give us files we can use in IGV. It's going to do so many things, but each one of these things typically had to be done separately previously. So now we have this container with the versions, everything being tracked, and a key that tells exactly how that process worked. And so we can see as this thing keeps going, we've already done the check design and done fast QC, and essentially we're done. That will not run as fast for you. I already pre-ran this to make sure it worked, but you'll see these things filling in time. And with this design file and these URLs, you will have your first ChIP-seq results. And we'll go over how to what all these processes are next time. For now, we just wanted to focus on installing Nextflow, installing a pipeline. This would be the same for RNA-seq, ATAC-seq, anything. A little bit different markdown, but then you just go read the instructions and follow the same process. So congratulations, you're running ChIP-seq, and we'll talk about all of these processes and the output files that you got in the next class. Until then, code light.